Hey, hi, good morning, everyone. So today is our last lecture of new material for this semester. All right, so it's hard to believe that we've reached the end of our course with today's lecture. Um, as a reminder, Monday we will have class, but it'll be a review session. So I will recap and help go over with you the content that you'll be responsible for next week's third exam. And then next week, Wednesday, will be the start of our third exam. You'll have your multiple choice on Wednesday. It'll be just like the previous exams where you just log into Laulima and take care of taking your multiple choice exam on Wednesday. And then in Thursday or Friday of next week in your recitation will be the free response. Okay, and then that'll be the last time you get to spend time with your TI, right? So hopefully, you know, you can express some appreciation for the hard work they've put in helping you all semester long. All right. All right, so let's wrap up our semester talking about segregation of alleles and recombination, and let's try to kind of step back and put a lot of these things together and get us thinking about, what this really means in terms of what we've been talking about all semester long, cell biology, and what life is. Right? So this is a slide that I just kind of threw together this morning, so it's not part of the PDF that you have, but that's okay, because I was thinking about this morning. One thing that I always, kind of amuse myself thinking about are interesting kind of situations where things kind of are, how should I say it, looping in kind of funny or strange ways. For example, I was thinking about small businesses that paint signs, right? And these businesses, if they're traditional brick and mortar businesses, oftentimes outside of their storefront, they would have, of course, a sign advertising that they are sign painters. And so if you think about this, it's an interesting situation, right? Because the sign that they paint to put outside of their front door to advertise that they are sign painters tells a lot about what they do, how they do it, how well they do it, right? And it has to attract customers. So here are some examples of signs that I just randomly found that advertise sign painters. Right? So these are hand signed. They all do sort of traditional hand painted signs. And so if you're in need of having a sign painted for you, and you go walking up or walking past these different storefronts, and this is what you see, you can learn a lot just by looking at this sign that's outside their door, right? This gives you an impression of the quality of the paints that they use. It gives you a sense of their aesthetic. What style of sign do they do, right? More traditional, more modern, and maybe it gives you some indication about, their business, how well they're doing, right? Because if you're a sign painting business, maybe you want to make sure that your sign outside of your business looks really good, right? So what does it mean if they allow their sign to get worn down, weathered, etc.? So in many ways, by looking at their sign, what you are seeing is the expression of many, many small different attributes or qualities about their business, right? And in the case of a sign painter, many of these things, you look at this and you sort of interpret and you get an impression of whether or not you want to give them your business, right? Whether their style fits what you're interested in, whether the appearance of the sign makes you comfortable that they are a reliable business and you want to actually invest your money in. Now, when we've been talking about expression of things throughout the semester, we've been talking about the expression of genes, right? Genes that are coded for by our genome, and each gene represents the blueprint, right? The recipe for a particular protein. And we've learned that the human body, the human genome, with its 3.2 billion base pairs spread out over 23 pairs of chromosomes, 23 pairs of DNA molecules that contain multiple genes along the length of the DNA strand, right? We said if we think about each one of those as a recipe for a protein, right, it's sort of an easier way of imagining being able to put everything together in one sort of volume of information, right? And that's why I was using our cookie book as an example. Remember, this is the cookie book that has sort of sentimental value to me because this is the Mrs. Fields cookie store that was next to the UC Berkeley campus that my wife and I used to go to when we were first dating, right? And then she bought a copy of this cookie recipe book, and to this day, she makes many of the wonderful cookies in it. So that's an easier way of thinking about this, because in the case of something like the cookie book, each individual set of instructions, each recipe, right, is like a gene that codes for a particular cookie, right? And so that's maybe a closer way of thinking about how our genome is, in a way, expressing, right, all these little attributes of what we have inside of us, what we carry, right, as part of our DNA. And so in many ways, what we see on the outside then when we look in the mirror is like one of these signs, right? Because it tells or expresses something about all the different proteins and versions, alleles, of these different genes that we carry, right? That determine things like obvious visible phenotypic traits, skin color, hair color, things like that, right? Height. But also think about this, inside of you, inside of the cell, we've been learning all semester, right? Proteins play important jobs, right? That are not just things that you see on the outside, but they determine how well things work on the inside, right? So even things that you don't see, like personality, right? Sense of humor, things like that, are all also attributes that in a way are influenced by our genes. And therefore, that's another way of advertising what genes we carry, right? All right, so let's kind of step back here and think about this back in the realm of talking about genes and our DNA. So the grandfather of modern genetics is Gregor Mendel, who was a Franciscan monk, a friar. He lived in the 1800s, and he was the first to really start introducing the concept of attributes or different traits of living things being passed on from generation to generation as discrete packets of information, right? Because prior to Mendel, people just thought that your different appearances and traits and qualities, whatever, your health, all had to do with just sort of more supernatural types of properties, right? And that your parents were just somehow part of just channeling these supernatural types of things and putting them into you as their child, right? So Mendel, his claim to fame was that he was the first to start doing real sort of genetic experiments. Of course, he did it with pea plants. And what he noticed is that if he had certain pea plants that had certain colored flowers, right? So the color of a flower is a very good example of some sort of visible attribute that has to do with the particular genetic makeup of the individual plant. He found that if he had purple flowering plants that always made purple flowers, right, and he crossed them with white colored flower plants that always made white petaled flowers, right? So he called these true breeding parents. In the F1 generation, the first set of seeds that he would get, and he'd plant, right, all the resulting plants would have purple flowers. Okay, so he realized that somehow the purple color is dominant, right? That somehow the white color is almost forgotten. But what's very interesting is Mendel took it a step further. He said, if you go ahead and cross this generation, these purple flowering plants, with other purple flowering plants from that same generation, what he saw was that if you had 4 offspring, right, for every 4, 3 of them would be purple flowering, but suddenly, amazingly, the white color reappeared, right, in 1 out of the 4. And so this, to him, meant that the trait was never lost. It was just somehow hidden by other traits. So this is the mechanism of what Mendel was looking at. We, of course, now know, right, you taking this course, now know far more about genetics than Gregor Mendel ever knew, right? In fact, it didn't even occur to him to consider calling this field of science genetics, right? But let's say here is one of the chromosomes in the pea plant, right? And so the pea, of course, reproduces sexually, right? So that's why you're going to have two parental plants contributing their genomes to make an offspring plant. Let's say the blue chromosome in this particular plant comes from one parent, the pink one comes from the other parent. Right? Let us imagine that on this chromosome somewhere, there's a length of this DNA that represents a gene, right? A gene that in this case, if it is transcribed and translated, right, it makes an enzyme. If you look at the genetic sequence, perhaps in this A1 gene, right, one form of the gene has a C, a cytosine at this particular position along the chromosome. Right? And so its sequence is CTA, et cetera, et cetera, et cetera. And if this version of the gene is transcribed, and this becomes part of the triplet code that ultimately is going to select for what amino acid gets linked together by the ribosome to make this enzyme. Right? If you have a C at this position, then the right amino acid gets put there, so the enzyme folds up properly and it works. And in this case, this particular enzyme is part of a pathway in the plant cell that makes a particular accessory pigment molecule. Just like we learned about beta carotene or xanthophylls as accessory pigments when we were studying photosynthesis. Here's another example of one. This is anthocyanin. Right? So if you have the functional enzyme, the plant can make the anthocyanin, right? And as a result, the anthocyanin appears purple. And so that's why when you look at this plant, you get a purple one. However, imagine that the other copy of that chromosome that this pea plant got, that it got from its other parental plant, right? Within that gene, the same gene, but here there's an allelic variation, right? Instead of the C at this position, there's an A, right? So in this chromosome, it is ATA, et cetera, et cetera, et cetera. When this version of the gene gets transcribed and translated, right? Because instead of a CTA, you have an ATA at this position, maybe the wrong amino acid gets put or a different amino acid gets put there. And as a result, the enzyme does not fold up properly. And if it doesn't fold up properly, it doesn't work. Because remember, enzymes are evolved to be very specific in their shape so that they can bind the particular substrate that they're supposed to bind and catalyze whatever biological reaction it's meant to accelerate. Right? So in this case, you get no anthocyanin. That means if the individual plant we're talking about has one blue and one pink chromosome, you still get a purple flower. But if the individual plant got two pink chromosomes, right, then it cannot make any anthocyanin, and it just defaults to being a white color, right? So this is interesting. It's not that the recessive allele codes for a white pigment, right? In this case, it's just, it codes for a defective enzyme that cannot make the purple pigment. So many of these attributes that Mendel looked at, these are just some of them: flower color, seed shape, color of the pod, the shape, flower position, right? Do the flowers on the plant, are they high up? Are they high up on the stalk, or are they in the middle? And also the height. Right? So Mendel was able to at least figure out that this pattern of inheritance of dominant and recessive traits held true for many of these types of qualities that he studied. Right? But he also realized that many of these qualities were more complex. It wasn't simply a dominant negative situation. Many traits are far more complex. Now, these days, right, we actually know we've been able to map all of Gregor Mendel's pea flower, pea plant traits to the particular locations on the chromosome of the pea plant, right? So this is kind of amazing, right? I think this would have totally blown Gregor Mendel's mind, right, back in the 1800s. But you can see here, all of these now have been mapped to particular chromosomes and particular locations on the chromosomes of the pea plant, right? So in our systems, in our bodies, right, because we are also diploid and have one copy from mom and one copy from dad, many of the traits that we express do follow a dominant recessive type of pattern, just like the simple traits of purple versus white flower color. But many of the traits that we express are far more complicated, because many traits aren't just dependent upon if you have this version of a gene or this version. Many traits, things like height, for example, right? So height was one of the things that Mendel looked at, but he found that it was very difficult to actually figure out if there was a specific gene for that, right? Just like in our bodies, there are many genes that contribute overall to whether or not we tend to be tall or short, right? Things that have to do with our growth pattern, like of our bones, as well as things having to do with our metabolism, right? That influence how we utilize the nutrients that we eat. So genetic variation then, right? We were discussing this last time. In diploid organisms, organisms that reproduce sexually like we do, come from the genetic variations or polymorphisms and mutations that are passed down through our lineage. Right? The sets of genes that were passed down from each generation, right, that resulted in your grandparents, then your parents, and then you, right, are the current end of the line of, you know, all of these types of genetic variations. So remember, we've said mom, you know, mom here contributes a haploid genome through meiosis, right? So her diploid genome, as her oocytes are developing, the gametes, they eventually go through meiosis 1 and then meiosis 2 to be able to then divide the diploid genome in half. So she contributes one chromosome 1, right? So we'll just say this pink pool noodle is the chromosome 1 contributed by mom, right? Now remember, if this is the chromosome 1 contributed by mom, right, this might have been how the chromosome was in all of the cells of her body, right, with gene 1, gene 2, gene 3 on them, as shown on the cartoon on the slide here, that each codes for a particular protein. Right? And remember that during meiosis 2 in her body, though, even though all of the normal cells, the somatic cells in her body, right, don't do meiosis, they just always stick with this particular chromosome 1 that she inherited from her parents, right? The gametes do meiosis to try to recombine these. So she will have, of course, a chromosome 1 from her mom, a chromosome 1 from her dad. Right? And during meiosis, that's when you have the opportunity for crossing over, right? Remember, during meiosis 1, that's when you can actually make a new version of the chromosome, right? That still is chromosome 1, right? It's still human chromosome 1, and it still has gene 1, gene 2, and gene 3 in the exact right spot that they need to be, right, in order for this to work. But importantly, what? This chromosome now has gene 1 and gene 2 from her dad, right, recombined with gene 3 from her mom. And so as a result, right, you have genetic mixing and matching here. For convenience sake, we will say then that this now becomes, this whole thing becomes the pink chromosome 1 shown on the slide that she is now contributing, right, as part of her oocyte. And let's say because of her lineage, her family history, right, these are the polymorphisms or the allelic versions of these genes that she has. Right? Her gene 1 has a G at this position. Her gene 2 has a C at this position. Her gene 3 has a T in this position. Right? So she and her, husband here, right, when they conceive a child, the same thing holds true. On dad's side over here, he's contributing in his sperm a haploid set of the human genome. His contribution of chromosome 1 likewise is a recombined version that he got from his mom and his dad that have been now mixed around during meiosis 1. He contributes slight genetic variations, right? His version of gene 1 has a T in this position, an A in this position in gene 2, and a G at this position in gene 3, right? And together, when sperm and egg come together, right, that creates their child, right? Okay, so I guess this wasn't chromosome 1. It's actually, I put the blue box around chromosome 2. All right, so whatever. It's chromosome 2 that we're talking about, right? So now you can see that's why their son's version of these chromosomes and the genes that he got, right, are actually different than the ones in the normal somatic body cells of mom and dad, right? And this makes him a unique individual. Think about this, right? We were talking about the pea plants and how different enzymes, if you have one DNA base different, can make the difference between if the enzyme, the resulting protein product of that enzyme works well or doesn't work, right? That might very well hold true for these 3 particular proteins that are coded for by these 3 genes as well. Who knows what impact having these particular genetic variations might have on the functioning of these fully made, folded up proteins, right? Maybe some of these work better because of the genetic variation. Maybe some of these don't work as well. And if this is happening with all the genes, the 20 to 25,000 genes, right, that are making you, then the end result is this wonderfully complicated expression of revealing what those different attributes are that are coded for by the particular genetic variations that you carry, right? You are the hand-painted sign advertising, right, the different traits that are encoded for in your genes. So just as a quick recap, so recombination then, recombination dramatically raises genetic diversity. You can already imagine that. If this type of crossing over is happening during meiosis 1 for, you know, all of your chromosomes, right? This is just another way of looking at what we looked at last time. Here during prophase 1 of meiosis 1 is when you have these tetrads forming, right? So dad's contribution, so the genes that you inherited from dad, right? That chromosome has duplicated during S phase, but the sister chromatids are still together. The chromosome that you inherited from mom, same thing, but during meiosis, so this is only happening in those special cells of your body, the gametes, right, or the cells that are making the gametes, whether the sperm and the egg, or the egg, right? This is where the crossing over can take place to have an opportunity to now mix these different genetic backgrounds, right? So this is meiosis 1. And so here is actually an illustration of this happening, right? So this is an actual image of crossing over taking place between 2 homologous chromosome pairs. Importantly, I think it's critical for you to remember, right, the difference between meiosis 1 and meiosis 2. Meiosis 1 is when the recombination takes place, and then meiosis 2 is after the recombination has taken place, right? Now you need to basically divide up the diploid genome into separate haploid gametes, right? So here's another way of kind of imagining this. If we were to pretend, again, for convenience sake, that the whole human genome just has 1 long chromosome and 1 short chromosome, right? Then at the start of your gamete-producing cells, undergoing the process of becoming, let's say in this illustration here, sperm cells, right? You would have a diploid cell to start with. You have the chromosome 1 that came from dad, the chromosome 1 that came from mom, the small chromosome, maybe this is like, you know, chromosome, whatever, like 21 or something from dad, and the one that came from mom, right? So for illustration purposes, let's just follow just the long chromosome over here, right? So you think about it. Your grandparents on both sides, right, contributed chromosome 1s to make your mom and to make your dad. Right? During meiosis too is when things got split up to make the sperm cells. Right? So mom and dad has a very different makeup than their parents. That's why they're not identical to their parents. Likewise, right, this chromosome 1 that was passed on from your grandparents, right, it's going to undergo meiosis 1 in mom and dad to produce gametes again, and therefore, right, their kid, right, is a recombined version of those. So one way I always sort of go back to thinking about then is, if we think about then all of the genes on our 23 pairs of chromosomes as being discrete packets of information. Each gene is a set of instructions or a recipe, right, to code for a particular protein. Right? And if we think about this back to our example of the cookie book recipe or the cookie book volume, we can say the cookie book represents an entire genome. But, one thing that I want to introduce now to you is, think about this, right? The cells in your body don't just have one copy of the cookie book. Let's say the cookie book came from mom, right, in the slide here, but what? You also have another copy of the same cookie book that came from dad. And check this out. It's slightly different, right? It's the same cookie book, but it's going to have slight differences due to those genetic variations, right? So this original Mrs. Fields cookie book, I think this volume was from like the early 90s, right? Because this is when I was in college, and my then girlfriend, now wife, right? This is when we went to the cookie store and bought this. So over the intervening years, this cookie book has been tremendously popular, so that they have a different edition of it now. But what's interesting is it's essentially the same cookie book, right? Because it's all of the most popular recipes, but there are slight differences. For example, if I were to go to, we were using the chocolate chip cookie recipe, right? This is what I was using at the start of the semester to illustrate the concept of this being a gene, right, that ultimately is going to result in the production of a finished product, the protein, right, or the cookie, sorry, right? And I made the point to you that, the cookie recipe doesn't do you much good. What you really want and care about is the finished cookie, right? So you have to transcribe this and then translate it to get your finished cookie. Of course, the later version of the book also has the chocolate chip cookie recipe. And, it's pretty much the same as the one in the older volume, right? If you look at it, they use the same pictures. The layout's a little different of the page. But importantly, if you look at the text closely, there are some differences. In the old version of the cookie book, it says, for example, drop the dough in rounded tablespoons on the ungreased cookie sheet, bake for 22 to 24 minutes, right? 22 to 24 minutes. Whereas in this version, the later version, I think this was published maybe like 5 years later, right? The recipe looks very similar, except it says bake for 18 to 22 minutes. That's interesting, right? This one says bake it for 18 minutes. This one says bake it up to 24 minutes. I wonder why. What probably happened is, over time, people's tastes in cookies changed. Maybe in the late 80s, early 90s, people liked hard, crunchy chocolate chip cookies, right? Whereas by the time you got into the 2000s and beyond, people are more into the sort of intentionally undercooked, doughy kind of cookie, right? Right? So by having both of these at your disposal, right, you have the capability of making both versions of the cookie, a hard, crunchy one or a softer, doughy one. And the softer, doughy one, right, kind of reflects a change in selective pressures, right? As more people liked soft, doughy cookies, it made sense for Mrs. Fields to modify her recipe so that her award-winning chocolate chip cookies would now be kind of, more doughy and chewy, right? So think about this also. On Amazon at any given time, there are something like 11,000 different cookie recipe books that you can buy, right? And many of these, I think just come and go, right? But the fact that Mrs. Fields' cookie book has lasted for something like 30 years tells you something, right? Because it's still ranked among the top hits on Amazon. What does that mean? That means this set of cookie recipes that Mrs. Fields worked on and came up with, right, have a lot of lasting power. Because time and time again, when people want to bake cookies, they're drawn, right, to these recipes. So even though, I looked online on Amazon, you can buy a cookie book that's all like vegan cookies, right, or something like that. There are a number of those. There are even cookie books that are for, cookie treats for your pet dog, right? Those, I think, unfortunately, maybe they're published one year and then that's it. They have a short production run, and that's, 10 years from now, you wouldn't find them, those same ones available. Whereas decades later, you can still find the Mrs. Fields cookie book, because those recipes, right, have stood the test of time, the selective pressures of us, what we look for in a cookie, we find, right? And think about this. If you were to make every recipe in this book and make a big buffet of cookies to choose from, right, that is essentially what goes on inside of you, right? You have 20 to 25,000 cookie recipes represented by your genes, right? And you make those recipes into you. Okay. So let's shift gears a little bit, right? And even think about this idea of a diploid genome. So hopefully it's pretty obvious to you from our discussion why having a diploid genome is evolutionarily very advantageous, right? Because it allows the mixing of slightly different versions of genes. And it's like Mother Nature's experiment, right? What alleles, what versions of these genes will stand the test of time based upon changing factors around us as a species, right? In the same way that from the 80s to the 2000s, people have now preferred to like soft, doughy, undercooked cookies, right? The changing world around us applies pressures as well. And certain genetic variations are selected for over time. And that's how species evolve. But consider this. Let's rewind all the way to the beginning of life on Earth, 3.5 billion years ago, when you had the first cells, right? Remember, life on Earth is all based upon having a discrete unit of life that we call the cell, right? Those first cells were not diploid or polyploid or whatever, right? They were haploid. Just the fact that you could string together DNA to make a set of genes that would work as the set of recipes to make proteins that will allow the cell to actually persist and carry out all of its living functions as well as reproduce. I mean, just having one copy of that genome, I think obviously, right, it makes sense that life started with a haploid a genome in organisms. So where did diploid genomes come from? How did that happen, right? So let's look at this. 3.5 billion years ago, bacteria and other prokaryotes, were haploid. They were one end, and they just reproduced by binary fission. When the time came, they just split, right? And you end up with two cells that are both one end. Right? So here, right, if you look at the whole life cycle, let's pretend that each circle here is like the full cycle between cell divisions. So the organism is one end. Right? At a certain point, it's going to go all the way around here, carry out all of its normal functions. And when it's time to divide, it's going to make a copy of its genome. So temporarily, it's 2N, but just as part of this dividing process, right? And then each of those 1N just goes to making a daughter cell. Right, so one daughter cell goes off to the right in this cartoon over here. The other daughter cell, we can continue following, right? It becomes now what we're following in this circle, right? So if you think about it, I already challenged you to think about this. If an amoeba divides like this, is an amoeba immortal, right? Is life an unbroken chain? Because if each daughter cell came from a parental cell, and the parental cell itself was once a daughter cell that came from a parental cell, right? You can go all the way back, right, through the history of life on Earth. So is life an unbroken chain from 3.5 billion years ago to today? If you're talking about an amoeba or even simpler, like prokaryotic organisms like bacteria, right? Let's now fast forward, right, from 3.5 billion years ago. Now we're looking at 1.2 billion years ago. So at this point, now we have eukaryotic cells, right? Cells that have organelles in them. We now understand that those organelles probably came from those simple prokaryotic cells, sort of taking in other simple prokaryotic cells, and they existed happily together in this kind of symbiotic relationship to create primitive eukaryotic cells, right? And over time, those little prokaryotic cells that lived inside, they lost their ability to truly function as a separate living thing. They became entirely dependent upon the larger prokaryotic cell that held them. And likewise, the larger prokaryotic cell became dependent upon the different processes that the little prokaryotic cells inside of them carry out, like things like mitochondria, right? That became the basis of aerobic respiration in eukaryotes, right? So about 1.2 billion years ago, early photosynthetic eukaryotes, such as mosses, began to alternate between 1N haploid life stages and 2N diploid life stages. So to this day, when you see a moss growing on a rock, maybe by a stream or something like that, I think all of us are familiar, that a moss kind of looks like this. Sometimes you'll see these things sticking out of them, right? And you might think like, oh yeah, the moss is like flowering or something like that. No, right? Technically, these are not flowers, because flowering plants came much later, hundreds of millions of years later. What you're actually seeing are the two different life stages of the moss, the 1N and the 2N. Right? So the let's see here. The one stage we call the gametophyte stage. This is the 1N stage. Right? The 1N stage, the gametophyte stage, is the part that you typically think of when you think of a moss. Right? That's the more sort of supple green part that looks a little bit leafy. Right? So it's called the gametophyte stage because it makes gametes. In other words, as it divides, it is 1N. All right? It only has one copy of the genome. So if it's able to go ahead and make additional cells that are 1N also, right, through mitosis, you make more 1N cells, these 1N cells can then go ahead and join up with other 1N cells from the same or other moss individual to form a 2N cell. Right? And so this 2N cell is what we call the sporophyte stage. So that's actually the part that sticks up here, right? And you have this capsule. Okay? So this is actually two different life stages of this moss plant. That 2N stage, in order now to divide, it undergoes meiosis, right? So that there's the ability to have some crossing over, and it generates 1N cells. These 1N cells then will fall off and begin to grow as another 1N gametophyte stage. Right? So there's this alternation of 1N and 2N life stages with something even as primitive as a moss 1.2 billion years ago. So when you think about this, is the life of a moss also an unbroken chain, right? If one life stage, the products of one life stage just become the basis of another life stage, and so on and so forth in this cycle, right? How far back do you have to go when you look at that little clump of moss on a rock? How far back in time do you go to find when the life of that particular clump of moss started? How far in the future do you have to go before the life of that clump of moss on the rock ends? Of course, barring dramatic changes, right? If you go ahead and you take that moss off the rock away from the stream and you lay it on the sidewalk in the baking sun, yeah, you could say, hey, I discovered the end of the life of that moss, right? But I think you know what I'm saying. If all things are allowed to continue going this way, right, is life an unbroken chain? So modern plants that developed, millions of years later after the mosses, they still follow this alternating 1N, 2M life cycle. So let's think about something like a fern. Right here in Hawaii, we really enjoy the sight of ferns by the side of the road, things like that in our yard. When I was growing up as a kid in Massachusetts, yeah, we had some ferns, but you had to go into the woods pretty deep and like find them and stuff like that. I always thought it was cool to find a fern because it reminded me of like, oh, this is like what the dinosaurs probably, had around them or something like that. But modern plants still alternate between this 1N, 2N life cycle. Right? So consider that meiosis evolved to give rise in plants to a pretty long 1N gametophyte generation. Right? So here you have spores. The spores grow into this funny structure, right? So this funny structure is the mature gametophyte life stage of a moss. Right? Then what we're probably used to seeing, though, is when we think of a fern, we're used to thinking about these very leafy fern-like structures, right? These are actually the 2N life cycle of the fern, right? Or the sporophyte. What you usually don't see is if you see a bunch of ferns growing, on the ground, right, is you don't realize that at the base of these leafy ferns, you can often find this funny little flat structure, which is the haploid or 1N gametophyte stage, life stage of the fern, right? So that's this funny kind of thing here that's on the ground, right? So within this one individual, parts of this gametophyte through mitosis, because it's 1N to start with, right? So as it just replicates its DNA and cells divide, you're making more 1N cells, right? It can make the equivalent of sperm and egg, right, on the same individual. Right? So that means a lot of times the sperm from the same individual fertilizes the egg from the same individual, right? And you get now a 2N zygote that then grows as this nice leafy fern that we're used to seeing, the sporophyte, right? And then the sporophyte then, when the time comes, it does meiosis to kind of mix the genes, right, and make spores, right? And those spores scatter. So that's like when you look on the underside of a fern leaf, sometimes you see those little brown fuzzy dots, right? Those are the spores or the sporangium that are going to disperse the spores. So you might say, well, that doesn't make sense because you don't get much genetic diversity there if in reality the sperm from the same individual plant is fertilizing the egg. So what's the evolutionary advantage of that? Think of this though, right? On the floor of the forest floor, right, the ground, you'd have many of these gametophyte individuals growing. And if you have rain, if you have puddles of water that cover them, so they're really damp and moist, right, then you'd have the opportunity for the sperm of one individual gametophyte to actually float and maybe make it, right, to another gametophyte that's nearby. So you actually would have the opportunity now of plants of different genetic backgrounds to mix, right? Right. So this worked, right? This worked, and this worked well for plants because it allowed them then to have a diploid genome that allowed them to have genetic diversity, right? And so you begin to sense that life, from these primitive steps began to head towards things like sexual reproduction. Even flowering plants to this day, right? You say, well, like, yeah, ferns are modern, but they've been around, right, for hundreds of millions of years. What about modern plants? For example, this nice tiger lily here, right? You're like, they don't still do this, do they? Yes, they do, right? But you say, wait, I'm pretty sure that this nice lily plant that I have growing in a pot doesn't have on its bottom some sort of weird, mysterious second life phase growing in the soil, right? That's right, it doesn't. Right? That's because the second life phase or stage of modern flowering plants, right, is just contained in the flower itself. In other words, Most of the life part of the plant is the 2N sporophyte stage. Okay, so that's when the plant is growing from a seed, and then eventually it has a flower, right? The flowering part is actually the 1N stage of the plant now, right? So these tissues within the flower are 1N, right? And they will then produce these gametophytes, the pollen, right, and the egg. And likewise, I think that's why many flowering plants can self-fertilize, right? But of course, what they're hoping for is that something like a bee or other insect will come and take the pollen from this particular flower and take it to some other flower on a different plant, right? So it's fascinating to think that modern flowering plants still do this alternation of generations. It's just one of the generations, the haploid generation, has really shrunk down to the point where it really only exists as the tissues that make up the flower, right? So in fact, the alternating life stages was staring you right in the face all along, because it's the flower, right, that you admire, and you realize that is the alternate life stage of the plant. So look on the circle here, right? So most of the life now of the plant is spent in the 2N stage, but when it comes time, right, to reproduce, it makes a flower, and the tissues in the flower are the gametophyte stage, right? They are 1N, and they make, through mitosis, 1N gametes. Right. So as life on Earth evolved, right, now we obviously did not evolve from plants, right, because we were separated from photosynthetic organisms a long time ago. But you can imagine, right, that Mother Nature was doing similar experiments like this throughout the eons of evolution, whether you're talking about plants or animals, right? So the same types of discoveries of what works well in terms of ploidy, right, holds true as animals evolved as well as the plants. So we said plants evolved so that now most of their life cycle is spent in the 2N stage, and they only make a 1N life cycle when it's time to make their gametes, right? So think about this. In our bodies, right, we still have this 2N life stage. I mean, that's you and I sitting here right now, right? The cells in our body are 2N. So what happened to our 1N life stage, right? What happened was Mother Nature realized there's no reason to have a completely separate life stage, right? If the whole purpose of that 1N life stage, the gametophyte stage, was just to do mitosis so that you can take 1N cells and make more 1N cells, right? So over evolutionary time, in animals, for example, right, that separate stage became so minuscule or so shortened that it essentially ceases to exist. So we now, right, skip a separate life stage. We directly make our 1N gametes from our 2N cells, right? So there's no separation of generations between 1N and 2N. The 1N just comes directly from our 2N through meiosis, right? So that's what our bodies do today. Higher animals use meiosis to make our gametes directly, right? So it eliminates this separate stage, but you can still think about it this way, right? Our entire lives, from fertilization, development, birth, and we grow up to adulthood, is our 2N stage, right? But inside of our bodies, these specialized tissues in our bodies, our ovaries and testes, right? This is where meiosis happens. And rather than having a separate 1N life stage, we just directly make our 1N gametes from those specialized 2N cells in our reproductive tissues, right? But think about this. Our gametes, the egg, the sperm, they're alive, right? They came from the division of living cells, right? So in a way, they can still be thought of as representing the 1N stage of human life, right? And then that stage is short-lived, though, because once they come together, they start the 2N stage. So in some sort of amazing way, we still see this theme of how life originally began on Earth and the experimentations of a haploid genome becoming diploid. So I pose the same question to you as I did with the prokaryotes, the mosses, the ferns, the flowering plants, right? Is life an unbroken chain then? Where does life begin? Where does life end? We conveniently think of our lives as being when you have an individual and you're born and you grow up and you eventually die, right? But if we reproduce and have kids, right, those living cells that represent our one end stage are passed on. So again, if you go back in time far enough, when did your life really begin, right? And when does your life really end? Okay, so I'm going to wrap up here because I know we're over time, but hey, since this is our last lecture, I think I kind of deserve to talk a little bit more, right? So over the course of the semester, we've studied, all of these wonderful molecular components that make up a living cell, everything from nucleotides that make up nucleic acids, that make up our DNA, our genetic sequence, to things like simple organic compounds that are put together to make more complex organic compounds, things like carbohydrates made from sugars, things like lipids that make up the fats that are so important for our cell membranes, right? And of course, amino acids that are put together in the order that's specified by those cookie recipes, right? By our genes that specify what order of amino acids should be put together to make complex proteins, proteins that are structurally important, proteins that are also functioning as small little machines to make everything else happen inside of our living cell. Right? Ultimately, though, life on Earth, all life on Earth follows this pattern, right? Starting from DNA or RNA, right? And all of this life on Earth, of course, as we've learned, right, is a result of the continuous energy that rains down on our planet from our sun, right, that fuels everything. It is the sun's energy that gives you the energy right now to be able to sit and listen to this lecture, right? That allows you to have thoughts and feelings and be able to do all the things that you do. It is the energy of the sun passing through all of the different life on Earth, from photosynthetic all the way up to animals like us, right? But of course, another important theme in this class has been what? The importance of water, right? Life on Earth is what it is because we are a water planet, right? 70% of the Earth's surface is liquid H2O. Earth is the right distance away from the sun that it's not too hot, not too cold, so that you can have liquid water oceans. And life began 3.5 billion years ago in water. And all those water molecules defined how those other organic molecules could behave, what their shapes could be because they are surrounded by water, right? And how reactions could happen to allow complex molecules to be made from simpler ones, right? Life on other planets that aren't water planets, don't have liquid water, I'm sure, absolutely sure there's life on other planets, but it's not going to look like us, and it's not going to use DNA, RNA, and amino acids the same way that we do. Remember, those molecules certainly exist, but they're going to be differently shaped, because if you don't have them surrounded by water, then those same biochemical processes that we rely on and all life on Earth relies on, right, wouldn't be the same set of rules on another planet. Right? That's why I said, we are linked to every other living thing on our planet because of water and how we evolved. And that's why I said, even a lowly little slug that's crossing the sidewalk in front of you, right, you are more closely related to that slug than you are to any other life form, right, in the universe. So I just wanted to end with this thought. When I was a little kid, one of my heroes was Carl Sagan. So Carl Sagan was a very famous scientist. He was loved and endeared because of his ability to actually bring science from the heady sort of academic halls of universities and things like that to the general public. right? So in 1982, I think, Sagan teamed up with PBS and made this a 13-part series called Cosmos, which explored many of these concepts of what we are, where we come from, right? And what makes us, these living beings in a universe, right? And as a little kid, I was blown away by that. Not only because I liked the things that he talked about, even though I couldn't understand it, but more importantly, I loved the way that he was able to kind of get a kid like me really excited. Right? And one of Sagan's famous sort of thoughts was that, we are all built of atoms, right? And the atoms date back to the birth of the universe some 13 billion years ago. And everything is made in the universe of star stuff, as he called it, right? The basic ingredients. And what I want to challenge you to think about is this. If life on Earth started 3.5 billion years ago, and we have this link, like this unbroken link to the first life on Earth that sort of invented and came upon all of these mechanisms for life to happen, DNA, proteins, lipid membranes, all these things that we've discussed. I want you to think about this. You and I sitting here right now, right, as part of the human species that has been around for maybe at most, maybe a couple million years, right? A couple million years in the history of 3.5 billion years of life on Earth. Right? That is a little blip in time. So it's very easy for us to get caught up in our everyday sort of challenges. We worry about things like our phone. We worry about things like not getting along with people. We worry about school. We worry about work. Right? A lot of times we feel like we're not that special, but we are special. Why? Right? Because you and I sitting here right now represent the first time in the 3.5 billion year old history of cells on Earth that cells are coming together and have the ability to actually understand what they are. Right? Not only what they are, but where we've come from, and really importantly, where we're going in evolution. And I think that is a wonderful thought. And I want to leave you with that thought because I hope this course has given you, beyond just knowledge and information about molecular cell biology, but a bigger appreciation for why molecular cell biology is so important and so relevant for understanding, our lives and making a difference. Okay. All right, so I thank you for a wonderful semester. And so I'm going to stop there today, and I'll see you again on Monday when we review for the exam. Okay? All right, bye-bye.